Good evening, everyone. Myself, Divya. And so, before I start, I'm thoroughly pleased to witness you all here in this free online seminar series on research and career journey in bioinformatics, which is a research seminar. And it's my privilege to address you all on the behalf of our organization, Dollar Education. I welcome you all the participants, and we are truly delighted with their presence.
We, as a company, aim to achieve great heights. Our organization, Dollar Education, is an initiative to offer practical trainings on emerging concepts by making them affordable for all and developing a consistent environment of growing together. We want to promote bioinformatics and make everyone to know the significance in the field of biology. The concept is just to connect and share ideas amongst the group. We named our organization because of its multi-faced meaning. That is, it is the currency name in around 20 countries. It is popular as an affordable symbol and also as a variable in computer languages. We want our participants to get best out of our workshops. In our workshops and webinars, you can easily post your queries and you can make it interactive by replying to the questions. Also, we aim to provide the source of every information that we are providing with proper references. And it will be a next career jump for most of the students, and it will appreciate diversity and multiculturalism. Students can understand the fundamental of the field while researcher can apply their ideas to your projects.
So, as we know, the computational power increases and becomes more accessible. The age of bioinformatics will accelerate our ability to understand and tackle global challenges like never before. From discovering new antibiotics to fighting pandemics or making agriculture more sustainable, the promise is great and the applications are already rolling in. So, if we talk about bioinformatics, it's a broad scientific research field that combines biology, computer science, data science, mathematics, and statistics to drive the analysis, the vast amount of data that associated with modern biosciences. Someone once told me that it allows reproducibility, which is one of the strongest reasons for publishing scripts and pipelines openly. So, for our webinars, we are conducting this free seminar series on research and career journey in bioinformatics. So, for today's session, we have Dr. Laura Harris, who is a senior bioinformatician in Neuro 11, and her topic of discussion is meta-analysis approaches to NGS, SARS, and cancer data. She has completed her bachelor's of science in microbiology from Michigan State University and master's in microbiology from Michigan State University. Then she also pursued her master's in cellular and molecular biology from Michigan State University and completed her PhD in biomedical informatics from Rutgers University. She has gained a lot of experience during her career. She's an adjunct faculty in or scientific laboratory at the scientific laboratory coordinator, associate scientist at Bioport Corporation, scientific project associate at Pfizer, research associate at MBI International, medical writer in MMS, lead faculty area chair science, health in University of Phoenix, and now she's the senior bioinformatics scientist in NeuroX1 company. She has added a lot of honors and awards to her credit. She got the Teacher of the Month by Rasmussen College in December 2012 and Excellence in Teaching Award by Davenport University in August 2018. Now I would like ma'am to take the session further. And thank you so much for the invite, ma'am.
Okay, I think I am good. Unfortunately, I'm going to do this. Can everyone hear me and see my screen?
Yes, ma'am. It's audible and visible.
Thank you so much. So thank you for the introduction also. It's a pleasure to be here, and I look forward to sharing a little more detail about that journey on Thursday's talk. So hopefully you all can attend. But today I'll be talking about a research project that's been growing for a few years now that I've been working on. And so today my talk is entitled Bioinformatics Meta-Analysis Approach, Analyzing Next Generation Sequencing, SARS, and Cancer Data. And as mentioned earlier, I'm currently the senior bioinformatics scientist at NeuroX1. And if you need to reach out at any time, I have my email address, ostry@gmail.com. And the Python scripts around what I published and what I'll be talking about today are freely available. I do believe in open science. And so with that, my GitHub is here. So please reach out this way or via LinkedIn, with any questions. So this is just a presentation overview. I'm going to spend a couple minutes as an introduction to our topic. And then I'm going to get into the details of the approach and how it's applicable at different levels to see how cells function. So I do this using 3 examples. I'll start by looking at infectious disease and specifically gene expression meta-analysis of mRNA expression data as it affects SARS infection in the lungs. We'll do a brief background and significance, and then all of these subsections have effectively the same 3 aims. We will be identifying gene panels for identification. We'll then be verifying those gene panels in non-overlapping data sets. And then we'll be comparing those verified gene panels across different SARS infection strains, different cancers, etc. And we'll start by doing that at a gene level. We'll then move to a pathway level, and we'll keep in SARS, so in that way we're not switching topics on you. It's hard enough switching the analysis. And then we're going to end with cancer and looking at using the same type of approach with methylation data, and then a brief conclusion. So let's get started. My overall goal for this project, all these aims, is to figure out the molecular changes associated with disease mechanisms. And I'm going to do that using a computational approach of next generation sequencing data, with the idea that I want to identify potential targets that can be developed into therapeutics to cure or at least help those patients. These approaches are the first time a meta-analysis approach of gene, pathway, or methylation signatures have been used to identify these molecular changes. And I'll define signature in a minute. And as I mentioned, I'm specifically focusing on SARS infection and cancer, but you could theoretically apply this to other areas as well. So here I have from the World Health Organization, the global health estimates for death. You'll see along the bottom, number of deaths in the millions of people worldwide. What I'm pointing out here is that, yeah, you do have things like heart attack and stroke, but you get lower respiratory infection is a number 4 killer across the world. You have tracheal and bronchial and lung cancers at number 6. And then keep in mind that this data was collected for 2019. So as we all know well too much, in 2020, the COVID pandemic happened. And suddenly a SARS infection became one of these top killers. It's just not shown on this screen because of the date. So then the first part of this talk is looking at gene expression in SARS infection of lung tissue, both in human Petri cell cultures and in an in vitro mouse model. And if you want details for this study, it is published in Frontiers of Immunology from 2020. So let's talk about SARS viruses for a second, because I don't assume you were a virologist. I didn't start as a virologist. I actually started as a bacteriologist. But anyway, amazing how pandemics will shift your career. SARS viruses belong to the beta coronaviral family. Now, this is effectively the common cold, as we call it here in the United States. It's a viral family that's usually not life-threatening, causes some mild upper respiratory irritation for a couple of days. But over human history, we have identified 3 deadly strains of beta coronavirus. The first one was back in the early 2000s. That was the severe acute respiratory syndrome, or SARS. We're now going to call it SARS-CoV-1 because it's the first time we saw SARS go lethal. A few years after that, there was the Middle East, respiratory syndrome, or MERS. That was caused by MERS-CoV, which is very similar in genetics to SARS. And then we all know COVID was caused by SARS-CoV-2. And there's a lot of discussion waiting for SARS-CoV-3, which hopefully will happen later rather than sooner. Fortunately, the SARS-CoV-1 and the MERS infections, their outbreaks were contained through aggressive infection control, so they never reached pandemic status like the SARS-CoV-2 did. But fortunately, in the last few years, we've developed some successful therapeutics in terms of helping some patients, but not all. Vaccines are available in some areas, but not all. And so now we're downgrading the COVID-19 pandemic to an endemic. But there's still room for improvement for treatment options, particularly as we prepare for what seems to be the inevitable SARS-CoV-3 outbreak in the future. So what is gene expression? Making sure we're all on the same page with our biology terms. A gene is a piece of DNA that is the basic physical and functional unit of heredity. And so you're taught in biology, you have DNA in your nucleus. It's going to undergo a process called transcription to give you a messenger RNA that then undergoes translation to give you proteins, which are those functional units. So when I say mRNA gene expression, I'm talking about the process of producing the protein product from a gene instruction. Okay, so basically this process. I'm measuring the amount of mRNA that's available within the sample. And I'm doing that using either RNA sequencing or microarray data that is publicly available from the Gene Expression Omnibus, which is a knowledge base at the NCBI, National Center for Biotechnology Information here in the United States. So you collect your data, or from me, it's from the public repository. And the samples then are going to be RNA sample 1, say for an infected mouse, and then RNA sample 2, say from an uninfected mouse. The lab's going to convert that RNA that's extracted to complementary DNA, and it's going to tag the RNA samples based on a fluorescent tag to identify which samples they came from. In this example, red would be for sample 1, which is infected, and green would be for sample 2, which is uninfected. The samples are then mixed and they're put onto a computer chip. And then there's a process of hybridization that sticks the fluorescent DNA to probes that are placed in wells on this chip. The chip then is read by a laser computer and the intensity of the color is captured. So then you can see if it's brightly red, you're going to see expression in the infected, but not so much in the uninfected. If it's green, it's in the uninfected, but not the infected. And if it's a white or yellow, you've got some mix in between. So this technology allows basically a whole genome capture. So that you get gene expression data from Gene Expression Omnibus that can provide just a simultaneous profile of hundreds, not hundreds, but at least 50,000 genes at the same time for multiple samples. So it's great for differential gene expression analysis. So here's what the data will look like. You'll have a list of your genes. These could be probes or symbols or gene identifiers. Here I'm showing them as ensemble IDs. I've got some samples. So I'm showing you that my experimental samples are these 3 columns and my control samples are the blue columns here. Now that I have this data, I can do statistical analysis on it. So for example, a t-score. Now, keep in mind, most of you, if you've taken a basic statistics class in biology, are familiar with a p-value, a t-score p-value. But that p-value is based on an actual score that a computer looks up in nauseating tables that were created like 100 years ago. So then with that said, you can take that t-score and just ignore the p-value for now. The t-score can be used then to rank differential expression of each gene or probe, whatever that well is, between your samples, between your infected and non-infected in this example. And therefore you can come up with what's called a gene signature, which you'll see here on the right. So then the genes with a positive t-score are going to be red and they're going to be overexpressed genes in infected versus control. And if I have a gene that has a negative t-score, it's going to be at the blue end. And that means that that gene is overexpressed in control compared to the experimental. And then if it's in the middle, there's no real change between the two groups. So if I look at either end of the signature, I can then get an idea of what genes go up or go down in response to an infection or another disease condition. So a gene signature, by definition, is a list of genes ranked on differential expression as defined by an appropriate statistic. So here I'm using t-score because it's more robust. It gives you more statistical power with its standard deviation. Most people will use a log 2 fold change. It does matter what you use, but as long as you're ranking them using a method, that's what the point is. So once you have these gene signatures, you can then compare them. And so I did it this way. So I took this GSE 47960, which is a publicly available data set in Gene Expression Omnibus. It had 4 samples of infected human lung epithelial cells in a petri dish. And it had 3 samples of uninfected, or I'll call it mock infected, infected with a fake virus. I then make my signature. I take the top 500 genes and the bottom 500 genes, and I use these to define query gene sets. Now, usually you'll get query gene sets from someplace like an MSIG database or Gene Ontology or KEGG, but in this case, I'm creating my own from another signature. So in this case, I happened to find a super series in GEO. So these data sets were made by the same people under the same conditions. So I have a second set of mice that were, or sorry, not mice, Petri dishes that were infected and uninfected. So this I can keep as a reference signature and keep all the genes there. So then I'm going to go to gene set enrichment analysis, which is a really popular program by Broad Institute at MIT. And so the GSEA will create an enrichment plot, and this is what you see here. So the enrichment plot has your reference signature along the bottom, and right above it, you see black lines. Those black lines are matches between your query set and your signature. And if you have enrichment, you will see those black lines cluster at one end of the signature compared to the other end. So when I do this with my top 500 or my positive tail and my bottom 500 negative tail, compare those to my signature, you'll notice that positive tail matches, close, I say, to the positive tail of my reference signature. And the negative gene set will cluster at the negative end of my reference signature. And it will create what's called a normalized enrichment score with a p-value. So basically, it's calculating the enrichment score and goes up every time you have a match, goes down every time you have a miss, or where you don't have a match. You'll calculate that maximum score. Any genes contributing to that maximum score are then going to be leading edge genes. So these are the ones that are thought to have the most biological significance to contributing to your problem, because it's contributing to the maximum enrichment score. So I collect those leading edge genes and I create a panel with them. So in this case, I went from 500 genes down to 203 because those were that cluster. And in the negative, I went from 500 to 114. That's that cluster. Okay. And I can look at the genes in those panels and figure out what they are. I can use the panels as a whole to look at other signatures, use them as query sets for other signatures. And then that could be used for verification and for comparison purposes. And then, of course, looking at the leading edge genes that would result from these analyses. So just looking at those panels I defined, about 13 or almost 6% of the positive panel already had reported associations with SARS infection of human epithelial lung cells in a Petri dish. So this shows that my approach is able to detect things that were previously detected. I also find additional genes that were not identified. And so I have 12, around 5% of the panel had a zinc finger, and around 2% of the panel was involved in solute carrier family. If I look at the negative panel, none of the genes that I found had a prior association, but some of them did have that zinc finger and a solute carrier family. So these are some features that might be handy if you're thinking about drugs that might target a SARS infection. So next, I wanted to verify those panels that I defined. And so I was lucky that there was a third data set made by the same people under the same conditions. So this data set is my true verification data set. But then there are other data sets I was able to find also, but they had a slight variation where they're using human adenocarcinoma cells rather than human airway epithelial cells. So still from the lung tissue, just a different cell type, which shouldn't matter much, but might play a role. So I have these data sets, create all these signatures, run the same comparison. So here you'll see from my true verification data set that I get really good normalized enrichment score for both my panels. I'm getting good clustering. And on this right side here is a random model. So I wanted to ask the question, are these normalized enrichment scores that I'm achieving with my panel able to be reproduced if I just do a random model? So if I just take 114 genes out of this signature at random and use that as a query set, would it give me a similar enrichment score? And I did that 1,000 times in order to create a null distribution, which is what you're viewing here. And what you'll notice are the normalized enrichment scores I achieved with my randomly generated panels come nowhere close to where my actual normalized enrichment scores are for the panels I defined. So that's an extra level of verification that the panels are truly enriched between these signatures. When I look at my other verification data sets, the long adenocarcinoma cells, you'll notice the same pattern where the positive panel is positively enriched, the negative panel is negatively enriched, and my normalized enrichment scores are falling with, how to put it, outside or very close to the edge of my normalized distribution, my null distribution. So again, further verification that my panels are enriched. So then I looked at the leading edge genes. So basically collecting the genes from these analyses and then looking at who is similar, which genes are similar. I find 51 positive panel genes are shared across all 4 signatures. And 10 of those genes were already reported. Again, I'm able to detect both established and novel genes this way. For the negative panel, 22 of them are shared, but since we didn't see any that were any that were reported in the beginning, it's not a surprise that none of these are reported now. This leaves 63 verified genes that I found with no prior association to SARS. So I've done all this in a Petri dish, and I wanted to know how that would relate to an infected mouse. And so at this point, then I find a mouse data set that infects with the same strain, with the same age, etc. Use my panels against that signature. You'll notice that my positive panel has statistically significant enrichment, but my negative panel does not. And you'll notice the genes are not clustering at one end. They're kind of spread out, where they're kind of clustering at here. You see a black line, very dark there. When I look at the normal distribution, now I'm losing that negative panel, but my positive panel still is within the edge. Okay, so it is showing enrichment in mice. So I wanted to know what genes were shared between the human Petri dish and the mouse. And so I did this Venn diagram, and it's a little complicated simply because you'll see these areas where it says, I see SARS genes not in platform. And the reason these exist is because I wanted to make sure I wasn't losing genes from changing species. So for example, a human may have one type of gene, a mouse may have another type of gene. And so when we measure it, we're not measuring the same sequence on the chip. And therefore what pops up in the human might not pop up in the mouse. And it might not be the same way if it does. So we have to consider that. And I'm glad I did, because I found that I would have lost 2 genes if I hadn't done this analysis. So basically I have found 20 genes between the human and the mouse. Here are those 20. Here are all of the data sets looking at IC SARS, okay, infectious clone SARS. The black stars are a t-test p-value that's less than 0.05. That's the one you're used to. And then the red is your t-score, with the redder being a higher t-score. The black boxes mean they were not included in the platform. So these are genes that I would have lost, particularly looking across the mouse. But what you'll notice here is that no gene, okay, sorry, one gene, this XAF1, have black stars across all boxes. And this is the power of using gene set enrichment analysis over something like a t-test p-value or a fold change log2 value that's equal to 2, some arbitrary cutoff. Because the problem with things like Fisher's exact test that a lot of those differential gene expression analyses are based on is that they only consider a subset of genes, the ones that meet a criteria statistically. But biology is messy. And biology might have a gene that's relevant that has a p-value that's 0.055 that would be excluded by the old analysis, but is picked up here. And this is the power of this slide. Where you don't see stars would have been excluded with the old analysis, but with this analysis is included. So I'll double back for questions or what have you later. You'll see this again, but anyway. So now that I've thoroughly looked at IC SARS in human and in mouse, I've looked at 7 signatures. I wanted to compare those panels across SARS infection. So I started by exhausting all the data sets from the early 2000s. So there's the Urbani strain, the adapted MA15, which is a mouse adapted strain. There are some attenuated strains that were developed for vaccine development, but they never took off because funding wasn't there. Then there were some MERS strains, and of course, SARS-CoV-2. So it's a little hard to see because of all the signatures. I tried to color code them to make it easier. But what you should notice is for the positive panel, look at all the white stars and all the red. That tells you its normalized enrichment score is positive and that it is statistically significant by gene set enrichment analysis. There is one point I'd like to make with this, and that is that you do have a dose curve in here. So this GSE 33266, you'll notice has decreasing amount of inoculation dose. And so that becomes interesting because the 2 lowest inoculation doses were where we lost negative panel here. We also lose it with the random model on the right side. So I decided to exclude at least the bottom one because you'll see here it's not statistically significant by random model. But anyway, beyond that, there is a limit for the lab. Doing that old Venn diagram, this one on the right is the one you should focus on. It shows that I found these genes individually across all the SARS-1 strains, and that there were 5 genes that overlapped across all the SARS-1 strains. Here are those genes. They're all related in some way to immune response, and I'll talk about that in a second. But again, this is your T-score table, and then you can see that, there's a couple where you would catch them all. Actually, no, I'm not seeing any where you would catch them all. So you would not get any of these genes by traditional Fisher's exact test approach. Here we've got the MERS SARS-2, same idea where we've got all red in stars except for this last guy. And when I'm looking at the random model, okay, I'm outside, even this one's outside. So this blue panel messed with me. So I actually reached out to the authors of the paper that discussed this data set, and I asked them about it. And now that I remember it correctly, let me backtrack. That was an unpublished data set, but you can still get their information in Gene Expression Omnibus. And I asked them why I was seeing this. And what they told me was that this particular data set was unpublished because they didn't feel the RNA quality was sufficient. And therefore, I'm posting this, and we published it, just to point out that, yes, you can have an issue with our analysis if the RNA is not good enough quality. So just something to keep in mind. So this one gets thrown out. But regardless, I still see those same 5 genes that I saw in SARS SARS-CoV-1, they're in MERS, they're in SARS-CoV-2. And then here's your T table like you saw before. So over like 37 signatures, I think it was 37 or 39, I think it's 37. We saw the same 5 overexpressed interferon-related genes across all signatures, all positive leading edges. So this is really strong support for targeting interferon response therapeutically in SARS patients. There is some literature to support this conclusion. So we looked as a scientific community at things like interferon beta therapy on certain human airway epithelial cells that were infected with SARS-CoV-2. There's been some mixed literature reports for interferon therapy for COVID patients in terms of their success, but there's a lot of questions around the timing. When do we administer that therapy? Is it going to be injectable? Is it going to be oral? What type of interferon are we using? Are we using alpha, beta, gamma? So there's a lot of discussion about how best to approach it. But the idea is that the literature and the fact that people are clinically already going in that direction supports the idea that my computational approach here identifies useful potential targets. So for the sake of time, I'll address questions at the end, and I'll just continue on with the pathway activity approach. So here we're going to do the same thing with the same signatures, except now we're going to add a step. And that's going to allow us to take the gene signatures and the gene level view and look at it from a pathway view. And so this work has been published. You can find it here. But anyway, a pathway is a series of molecular actions in a cell. It's usually directed by individual genes in some sort of a progression that leads to a particular protein being made, a particular phenotype being displayed, etc. So the example here I just kind of lifted from my dissertation, has nothing to do with the research here. But anyway, you know, it's the aspartate to lysine pathway. Well, you start out with aspartate, it clicks into an aspartate kinase, that does a conformation change, which then creates an intermediate molecule, which goes to another protein, which then does another metabolic step, and it's a series, which then ends with the production of lysine, which then of course can inhibit the allosteric site of the aspartate kinase and shut the whole process down. This is a negative feedback inhibition loop, which you should have seen in some biology class somewhere. So this is just an example of one of the probably millions of pathways going on within a cell at any given time. And you can see how individual genes then can be collected to represent a pathway. So when I say pathway activity changes, now I'm measuring the expression changes for several genes in a pathway and calculating an enrichment score based on that. So I'm going to do that by taking all those gene signatures that I just made, and I'm going to compare them to 7,500-ish gene ontology biology process gene sets. Now, I got all these from the MSIG database from the Broad Institute at MIT. They also have them for KEGG. They have it for the MF, the molecular functions, the cellular compartments, all sorts of different things. So you can, get those gene sets, or you can, create your own, do whatever you want. But the idea here is that I'm taking known pathway gene sets, and then I'm going to rank each of those pathway gene sets by normalized enrichment score. And so now the pathways with a positive enrichment score, normalized enrichment score, are going to be towards the red end of my pathway signature. The blue end's going to be the ones that have a negative enrichment score. And then, of course, the ones in the middle have no change. So a pathway signature is a list of these pathways ranked by GSEA normalized enrichment score. And okay. So now that I have these pathway signatures, I can do the same approach I did before. I can use one of them as a query set, the other one as a reference, and run gene set enrichment analysis to get leading edge genes. Now, one of the critiques of the prior analysis, the gene expression analysis, was that I only did this one way. So they asked, did it change the results if we did it the other way? And if you read that paper, the short answer is no, it does not. But it did make me think about this a little differently, and maybe it would be more robust to then figure out which genes are, or sorry, which pathways or genes, works the same way, are the same between my two identification signatures, and define panels based on that. So when you go to my GitHub, that's the new way it's done. It's going to be a reciprocal, like what you see here, which is built upon the gene expression study. So in one area, I'm going to take one signature to define my query. And then for the second one, I'm going to flip them. I collect the positive and negative leading edge genes. The similarities become my panels. And the same thing can be done. I can identify pathways, verify the panels, use the panels as future query gene sets or panel sets. So here I've got those panels, 195 positive ones, 103 negative, 17 of the upregulated ones, so almost 9%, were previously reported. And quite a few of those came from the original paper. No surprise, one of them will be interferon. The downregulated pathways, no prior association. So then with the remaining pathways that had no prior association, quite a few of them are related to cellular processes. Quite a few of them are related to immune or viral response. 10% are decreased ciliary action, which isn't a surprise because in the clinic, patients with COVID are usually struggling to cough up phlegm. And then, of course, changes in nucleotide and protein production, which would go along with the viral response. Here are my verification data sets. Everything positive is red and starred. Everything negative is blue and starred. I need to point out quickly the range. Notice my normalized enrichment scores are huge. They're 10 and negative 10. So this is the random model on the right, and you'll notice this goes 3 to negative 3. So this is why you're not seeing the stars that I had on the prior gene expression. These values are so far outside the range that you wouldn't even be able to see the random model if I had done that. So be aware, the achieved normalized enrichment scores are much greater than what's being shown with the random model, which is good. That's what we expect. Looking across all those leading edges for the verified panels, okay, about 60% or so of the positive and around 30% or so of the negative were verified across all of them. So good reproducibility. Again, quite a few of these are viral defense, immune response, a couple of them related to circadian rhythms. All right, fine. Quite a few of them had no prior association. So again, finding things that are reported and things that are novel. Here's looking across all of those signatures for SARS-1, MERS, and SARS-2. I'm mushing them together instead of showing them individually because you should be used to seeing this by now. The red columns, red with a bunch of stars. And what's fun is even that one gene set that we struggled with, remember the one that had bad RNA, it even shows positive and statistically significant enrichment. So if we just take the analysis up to a higher level, so to speak, and view the cell activity from pathway rather than gene, we actually pick up signal and can include it in our analysis, which is pretty interesting. Again, these values are quite a bit outside what the normal distribution was, so that's why they're not being shown here. Doing that wonderful Venn diagram thing, you're going to see 11 overlapping pathways between SARS-1, MERS, and SARS-2. Here are those 11 pathways. You'll notice interferon is the top scorer via a Stouffer's z-score. That's not a surprise, since the 5 genes we found in the gene expression analysis were all related to type 1 interferon. But notice, using that pathway approach, we now pick up cytokines and CD4 helper T cell activities and a type 2 immune response. All things that were not picked up with the prior analysis that just looked at a gene level. So this gives you some idea that looking at different levels and using this approach to look at different levels helps with a whole cell theory kind of idea. These results are not surprising. There's a lot of literature that talks about the huge cytokine storm and the strong immune response that patients encounter, and frequently is lethal when we have a SARS infected patient. So, you know, the results are not surprising and very strong in terms of being able to use this approach. So here we started thinking outside the box. We started wondering if gene expression data, this whole messenger RNA, microRNA, hold on one second. Brain reset. So we wanted to ask the question, if we were limited to microarray and RNA-seq data, or if we could use other data from gene expression omnibus with this approach. And so this paper is hopefully going to come out within a couple of months. It's currently in review with, who are we with? I think it's PLOS Computational Bio. But anyway, so the idea here is to apply the same approach to methylation data sets. And in this case, cancer has a lot of methylation data sets available. And as I showed in the beginning, lung cancer is one of the top killers. So we decided to start there. So just a little bit of background on cancer. Cancer is a disease in which some of the body cells will grow uncontrollably and start spreading around. Causes around 10 million deaths in 2020 alone. And that's 1 in nearly 6 deaths. And that's saying something, considering that statistic came in the pandemic. Lung cancer is the leading cause. Around 25% of cancer deaths are lung cancer. People die of lung cancer more every year than colon, breast, and prostate combined. And almost all cancer cells possess some sort of a epigenetic abnormality. There's something wrong with their methylation pattern. And so that is why we decided to look at methylation patterning in cancers. So just a real brief thing on what methylation is. Methylation is the epigenetic mechanism that's using these histones to control a cell turning a gene on or off. And so you'll see here a normal cell will have various little methylation pattern that turns on some tumor suppressor genes. If we either under or hyper methylate this area, we can get gene stability or inactivity, which then turns off those tumor suppressor genes, and lo and behold, you get cancer. So when I say methylation changes, what we're doing is we're actually measuring the methylated cytosines between cancer and normal samples. And the methylated cytosine number then becomes representative of the status of the methylation. So how is this data generated? Well, in the lab, you basically have this bisulfate conversion, where anything that is not methylated and is a cytosine will be converted to uracil. Whereas if a cytosine is methylated, it stays unchanged. Once you do this conversion, then you can use PCR and some other quantitative things in our good old chip method to get data that's what you saw before. So methylation profiling data is looking at methylation patterns at a single base resolution across the genome. Your data looks remarkably similar. So I have my samples that are clustered based on my groups. Here I've got methylation sites, so these are sequences, but effectively that's what we had with probes on a microarray anyway. And I can rank the difference then and make a methylation signature, which is a list of genomic sites that are ranked on that differential methylation based on, in this case, t-score. Use this approach where I'm going to take some lung adenosine carcinoma versus some adjacent normal tissue samples. These are taken from the same patient, just cancer versus non-cancer part of the lung. Create signatures, do my analysis, come with my positive and negatives, figure out who is common, define my panels. So lung adenocarcinoma methylation panels. My positive one here has around 320 sites, and my negative has 304 sites. So then looking at those sites, most of them have been previously reported. Now, this is not a surprise because there's so much research going into cancer. So many people are doing so many different analyses in the lab, in the computer. Everyone's going cancer. So I'm not surprised. But the fact that I was able to find 11 sites that were not previously associated says something. And if I look at the negative panel, the same thing occurs. I find 17 probes that were not previously reported, though I do find quite a few ATPases and TNF receptor mediated apoptosis sites, et cetera, that were previously reported. This is just a table showing you the different groups with the different data sets that I used. These are the verification ones, so they're all using adenocarcinoma from lung. The only one to point out is this 49996 because it uses normal patients rather than adjacent. So these are actually different people rather than same person, different part of the lung. So that's why I labeled it as versus norm. But again, the idea is the same. I'm going to get positive enrichment for my positive panel, negative enrichment for my negative panel. Does not matter if I'm doing it with the normal people or the adjacent people, you know, you get the same thing. This is my normalized, sorry, not normalized, null distribution here. And you can see then I'm getting 4s that are well outside my range. So now that I've done this for lung cancer, adenocarcinoma, we want to spread out across cancers and see if we can find methylation sites that might be a global contribute to cancer. So I started with small cell lung cancer, went into glioblastoma, breast cancer, cervical cancer, prostate, bladder, oral squamous, gastric, colorectal, hepatocellular, and 2 types of adrenal cancers. This is the normal, null distribution, and you've got your normalized enrichment scores for the panel shown. You'll see that for the most part, you've got them well above or well below what you would get from the null distribution for the random model. This one is towards the edge, but still statistically significant. So then I want to know out of all these, which sites were common across the leading edges. And so for this, I decided to do a histogram just because the amount of signatures in a Venn diagram was getting nauseating. So what you have here is the frequency across leading edges. And these are the number of leading edge probes across those cancers. So I had 32 signatures total. So I allowed 1 miss. And I found that 10 positive probes and 6 negative probes were consistent across 31 or all 32 of those signatures you see here. So those are the sites that are most likely going to be the global methylation sites that we might want to look at. So here I've got my signatures. I've got those sites. I've converted them to gene symbols here simply to give your brain a break. You might recognize a couple of them like SOC17 where you're not going to recognize the probe symbol. But the point here is the arrows. You'll notice that this PRDM14 is my top score by the Stouffer's p-value. This DEFB 118 is my best for my negative. You would have thrown all of the if we had used a prior analysis of Fisher's exact kind of thing. It's found across all of these cancers. And so some of them had associations already with one or more type of cancer. So that PRDM14 I mentioned was already associated with pancreatic cancer, breast cancer, and leukemia. And they've been having issues with sensitivity with this particular target. But it wasn't found in other types of cancer. And so one thing that's interesting about this study is that these global mechanisms give a different perspective, where we might find these individually with certain types of cancers, but this gives you a more global type of approach across all the cancers. Okay, so our defensin beta 118 here was huge in gastric cancer, never studied before in lung cancer. Now it should be, according to this approach. That work will need to come now. So just remember, the overall goal was to try to cure disease using the computer, or at least predict some targets that we can take into a lab or a clinic to help people. And I think I've done a halfway decent job of demonstrating that this gene set enrichment analysis approach can be used at multiple levels to view how a cell functions and then to identify some of these targets. And so before I take questions with whatever time I have remaining, I don't really know what time it is anymore. It's been a good talk. I'd like to thank my people for help. So Amber and Marius helped me with the SARS paper. And then Radhiman and Tanman helped me with the cancer paper. All were volunteer students that were just awesome to work with. All right, so now I'll check the chat. I did see two up here. Okay. Oh, I only have one question. So hello. When we have to perform the functional annotation of differentially expressed genes, is it better to separate positive and negative genes into different groups? Oh, yes. I'm actually currently writing a paper that talks in part about that and how some people, they go for the p-value. And the p-value is going to mix your positive and negative genes. And you have to keep them separate because in a cell, the direction that goes matters. And for targeting purposes, the direction it goes matters. So if all my genes are interferon and they're positive, and I want to target interferon, I need to know that they were positive so I can then inhibit them to bring them back to normal. But if you're going by p-value, you lose the positive and negative, and then you don't know what direction you're supposed to take it. And then it also gets confusing for the analysis part of it also. So when you're doing the Fisher's exact test, it can confound your results. So yes, always keep them separate. The only time you would keep them together is when you're looking at the whole signature, in which case you're keeping all those middle genes too. You keep all the genes or all the pathways. That should have sufficiently answered your question. Let me know if you have a follow-up question, Caesar. Any other questions? Okay, here's another one. Glad that worked for you, Caesar. I am very new to bioinformatics from an IT background. Okay, cool. I love people crossing the aisle. Can you please let me know how do I go about it? Okay. As I'll express on Thursday, I'm actually coming from the other side of the aisle. I'm a trained biologist who has now transitioned into IT. So when I encounter people from your IT background, you all are awesome at programming. The things you can make a computer do just astonish me. But I find there is a definite lack of biology understanding, particularly when the papers are published. So you'll get like the paper that will say such and such was predicted by the computer. But then you look at what was predicted and you're like, there's no way that has any biological relevance. And I only know that because, you know, I've got 25 years of biology experience. So I would recommend partnering with someone that does have a biology background and really work with them to figure out what is it they're doing in the lab, what data are they generating, what can you do to advance that, and try to work together with the biologists to bridge that gap, because those are the best papers that I see, are the ones that have that interdisciplinary team. And if you need help finding biologists, you know, feel free to either contact me on LinkedIn, or you can go ahead and search through anything on my LinkedIn profile. I got a lot of biologists on there, but that's my recommendation. Find a biologist to collaborate with. Great question, by the way. Any other questions? All right, well, while you might be typing, I'll remind you that the scripts for these and example files are on GitHub. I use Python with Anaconda. If you are a biologist who is not used to programming, don't worry. I also have a repository of courses that will help you install Anaconda, helps you use a Jupyter notebook, all that good stuff. And of course, I'm available for questions. I'll go ahead and stop my screen share just so I can go find my LinkedIn page. Let me go find my LinkedIn page. I can throw that in the chat box, and you know, if there are no more questions, there you go. There's LinkedIn. Okay. Well, I don't know if there's anyone after me, but it's been a pleasure talking to you all, and I look forward to Thursday's talk when I get to share how I got here. It's been a journey.
Thank you very much. Thank you, Dr. Laura Harris. It was really a pleasure listening to your research. Now we are excited to listen to your career journey, and we all will be there on Thursday on the same timing. And thank you very much, all the participants. See you on Thursday again. Thank you once again, Dr. Harris.
Have a great day, all.
You too. Thank you.